Hey guys, so today we're talking plant viruses. I am not an expert in plant viruses, but they are super duper important, and so that's the first thing I want to address is the economic burden and their importance. It's tough to get a good synopsis of how much damage viruses cause to plants, but 30 billion is probably an underestimate. It kind of depends. So that's annually. So $30 billion annually, but that depends on specific situations. So, for example, I'm just going to take you through a few of these. So this is all from this same article. It's a really nice review article. I just grabbed one of their intro paragraphs, talked about a bunch of specific examples. They're the ones that estimate 30 billion, which they're referencing another reference. But so cassava, however you want to say it. So that is a huge problem. So that feeds a ton of people in Africa and India. And so there's cassava. I got a picture of each one. It matches up. This is the specific disease that it causes. And you'll notice a lot of similarities here too. You often see this kind of a mosaic-y thing. So tobacco mosaic virus was the first virus ever isolated because it was killing tobacco crops. And they have a lot of money, so they asked a bunch of scientists to figure out what's killing our crops. And that was like one of the first lectures we had was talking about how they isolated that using ceramic filters, right? And so in this case here we've got another mosaic virus. So we name these usually after their symptoms, which we do that in humans too. And so cassava, uh, the problem with that one is it feeds so many people. If you wipe out an entire set of crops for that year, you're going to starve potentially 500 million people, which would be really, really bad because then those people are going to depend on others, and either people are going to suffer, and other people's suffering makes you suffer too because you can't just sit idly by and let people suffer. And so we start giving them food, and that puts a burden on our economics and stuff. And so it's just a cascading effect. So getting rid of these problems is a big deal. Here's a potato leaf roll, see, because the leaves roll. Get it? Um, we're going to see all these names, begomoviruses and polaroviruses and closterovirus. I have a list later of just all these virus names. Most of them I've never even heard of because they're just not super well studied. Which kind of makes sense on one hand because we worry more about viruses that hurt us, our species, but these should be studied more because they're affecting us. I mean, $100 million, hundreds of millions in the U.S., millions and millions in the UK because they love potatoes there too. It can infect anything in that species or in that genus. Maybe it's family. So tomatoes are included in that. That's a ton of money. Citrus tree culture, so you can see that poor little citrus tree. Oh, he still made fruit, but it's not going to do super great. I bet you that fruit doesn't taste good. This one, the citrus tristeza clusterovirus, it estimated they lost 100 million trees worldwide of citruses. That's a lot. Here's barley yellow dwarf. So the barley yellow dwarf virus, see, it makes it yellow, and then the leaves are dwarfed, so they're a little smaller than they're supposed to be, and it's barley. So barley yellow dwarf. It's called the ludiovirus. Just in the United Kingdom, it costs $10 million a year. That's crazy. It's estimated. What is this? Charcot disease. So that'll affect all these rock fruits. So you've got like your or stone fruits. I'm sorry. So you got your plums, and your apricots, and your peaches, and things like that. There's something else. I just learned another one that's only in like India or something. I'll look it up later. 10 billion since the 70s. That's crazy. It affects the fruits, so the fruit don't look right. They probably don't taste right either, and the leaves don't look right. So man, it's really bad. Just a couple more here. Rice in general is a massive crop. Just in Southeast Asia, they lose billions every year to viral diseases. Just viral diseases. I mean, that's not including like funguses. Um, there's so many rice viruses. I just picked one, so the rice dwarf virus clearly is infecting this plant and not this one. Um, yeah, that's really interesting. And then this one was a relatively new one. So it's in Ghana, the cacao. So they're making like chocolate, basically. This cacao swollen shoot Badna virus. It's over in Africa there near Ghana where they grow a lot of those. And there's lots of, there's not a lot of pictures. There's a few. The quality is usually poor. And that's what's really interesting is we depend on cacao trees, like for food that we eat. And yet you can Google, I don't know, rice virus, and there's a million pictures. And you can Google, I don't know, this virus with the barley, and you'll get a million pictures. But this one, not so much, because people don't really care as much. They engaged in a massive culling of their cacao trees. Um, they said one of the the paper itself referenced another paper that said it was one of the most costly efforts by any country in the world to fight a viral disease. And so you can see they get these swollen like nodules here, and then the plants just don't really produce very well. Really interesting stuff. Massive, massive problems, and it's not just on the global scale, but it's more these local industries that get really damaged. So, like, we don't actually eat a lot of cassava in America, but a bunch of people all over the world depend on it, and so I don't really want to see them suffer either. So actually understanding viral diseases. So it's not just viral diseases. We can kind of lump other diseases in here against plants as well. But the key, and we're going to talk a lot about this, is the vectors. So plant viruses require a vector to enter. And so once you start dealing with, okay, we have this kind of plant infection, what do you recommend we actually do? And it can get really, really complicated because sometimes we buy seeds from other countries. We depend on different crops depending on a different country. We depend on your neighboring country could have different rules. You could be a farmer that has land in two different countries, right? You think some of these smaller countries where they like maybe you share land on both sides and they have different rules about like culling or incineration, or the EU has some really strict rules about GMO crops. Other countries not so much. And so like what rules are you actually following? Is your neighbor even following the rules? That's a great question. And so things get really complicated. And then the key here, so this is really talking about just all the different strategies they have. But down here is resistances and tilling. That would be like artificially generating mutants and or like figuring out what natural and artificial resistances actually exist genetically, and then using that to breed resistant cultivars. And so that's like GMO cropping, basically. Well, that's what it says, GMOs. But there's different ways to do that, which is really interesting. So how do we really assess that? Something that I wanted to bring up a lot was there's a reason that you want diverse plant cultures and you want your ecosystems to be diverse. Everybody's so obsessed with diversity now, but that's not that's not what I'm talking about. Diversity is super important, especially on a ecological level, because when, like, let's say you have only apple trees growing or whatever, or in my yard, I only have one kind of grass, and then all of a sudden there's some kind of pathogen that infects the grass. Now my entire crop is gone. And so that's why you want to kind of mix things up. Now with farming, it's a little trickier, but with um, like wild ecosystems, that's why we always want to have, a lot of diversity in those groups. There are municipal rules about that as well. I have a house, and I've read like a lot of the municipal rules for like planting trees in your yard, because I planted some trees the other day. And when you build a new house in Ozark, Missouri, where I live right now, um, they have specific rules. They'll have like, okay, here's 8 different tree types, and you can't plant all the exact same tree type in the same area, or you have to change things up, which is really, really interesting. So and you could say, well, why are you doing that? You're making it harder on the builder. Well, that's something that we've decided to make a rule about because it costs our community more money down the line if we only planted oak trees. Oh no, some kind of pest just killed all the trees. Now nobody wants to live here. Well, that's going to cost a lot more money. So we try and front end load our money and charge people. So that's kind of what this is talking about, is just the growth of the virus and the evolution of a virus in a specific community. And so you have either heterogeneous communities, which that would be like you have multiple plants growing in the same area versus a homogeneous one. And then the virus is actually is it going after? And then there's vector. We're going to talk about vectors in a minute. And then how does it actually evolve as far as genotypes go? And it depends on what kind of selective pressure is being applied. So the community is applying a selective pressure onto the virus, and the virus is applying a selective pressure onto the community. And this one's specifically talking about the virus being able to either manipulate the target host or minimize the a cross-host trade-off. So that's like if you have a really homogeneous community, it's not going to be very high. But if you have a heterogeneous community, then the virus kind of has to deal with that because it has to deal with multiple viruses. So it makes sense logically if you look at this, but basically viruses and plants are constantly affecting each other, and it depends on how they're getting transmitted. So we need to talk about transmission. Now, viral transmission is really, really interesting. So as I said, it's almost always vector driven. You need, just like in the last section, we talked about the last slide set, or the last presentation I gave, the last lecture, that's the word I'm looking for. We talked about human papillomavirus, which is a non-enveloped virus. Well, most plant viruses are non-enveloped because plants have a nice thick cell wall, which lets them be resistant to envelopes. They don't have a phospholipid bilayer sitting outside. They actually have a cell wall outside of their phospholipid bilayer. So you can't just attach to that cell wall and get in. You need something to actually push you in. And so that's where you're going to depend typically on an insect. So that's going to be your vector. It depends on the virus, what vector you use, and we're going to talk a little bit more in detail about specific vectors. But I really like this chart because it goes through like the complete order. So if you kind of have like your insect here is going to, all right, let's see right here. So let's start here. The insect inserts its mouth part or whatever and infects the cell types. It grows, and then another insect comes along and feeds on it. It grows inside the insect or just kind of hangs out there, and then eventually it will push it back into a new infected, an uninfected host, and then the cycle continues. Typical, I mean, of any kind of vector-driven viral paradigm. But it's really, really interesting in that you usually have these insects with specific mouth parts. The plant viruses are super diverse. Tobacco mosaic is the classic example of a really long helical, they could just be like massively long polymers of amino acids, proteins, but they can run the whole gamut. Notice there's no envelopes here though. Mosaic, see, so it makes leaves look mosaic-y. It makes these ones look mottled. This one, they look mosaic, yeah, some consistency there, right? There are lots of plant viruses. Most of these I have never heard of. Rhabdo I have, that's like rabies. Bunyiviridea I've heard of. Not much else on this chart. I am a human virologist, but this is super interesting, and as I said, it's very, very important. But a lot of these are really, really, really understudied, like hardcore understudied, even though they are super interesting and have super cool virologies. So this comes from Viral Zone, which is awesome. I like their diagrams because they're clickable. Not here because I just took the picture from there, but you can click on this if you go to the website right here. And you can click on it and follow, and you can see it kind of depends on mostly on how the insect is going to get it in there. Now, you could have an infected cell, and you could have the virus cross the plasmodesmata, which is their cell-to-cell contact between the cell walls. But ignoring that, or from like the xylem or phloem and get in. It's the same idea, though. But usually you're going to have injection from an insect because that's how you're going to get this initial cell infected anyway. Though we're going to talk about funguses and how they can do it too. And then it's just going to follow a typical viral entry. It could be an RNA virus, it could be a DNA virus, it doesn't really matter. But how it's getting in, and then it's just has to get inside and take over, right? And start making more viruses. And then hopefully if you're the virus, you want something to come along like this aphid looking thing. I think that's an aphid. I have a few of these pictures, okay? This one, the quality is a little lower, but it talks about, as a vector, I like this down here because I want to emphasize this big idea that you can either have circulative or non-circulative viruses. Usually when you're dealing with human vectors, so like think mosquito spreading dengue virus or West Nile virus, usually there's a life cycle within that vector host. So the mosquito itself becomes infected with the virus. In plant viruses, it can go either way. So like for those human viruses, the mosquito itself can't take a blood meal and then take a second blood meal and transmit the virus. That's pretty atypical. That's why we don't really see like if someone has HIV and they get bit by a mosquito and then the mosquito flies over and bites somebody else, that person's probably not going to get HIV. As a hypothetical, sure, but I don't think that's ever been documented. Because it turns out you need virus to exist in specific locations inside that vector host. And so in some plant viruses, we do see that where like an aphid or man, what are some of these other ones? There's a ton of different ones. Midges, I think grasshoppers can, um, but they just have to have like pokey mandibles usually. Um, and so the virus can circulate and actually infect the host, which is good for the virus in that it can make a lot more virions that can then get put out, which could increase the odds that it'll actually infect the host, a plant cell. But there are examples where, like, you could have this aphid take a meal from one plant cell and then move to a plant cell, just on the other side of the leaf, and just even if it's the virus is just inside its like little mouth parts there, it can still transmit the virus. And so that's just one example. And so there's a few different ways that we see different viruses exist here. And so, for example, this would be that example where it just kind of sits inside their mouth parts. So it's non-circulative because it's not circulating inside the insect, and it's non-persistent, so it's only there for a certain amount of time. You can have other ones where it kind of like sticks to the mouth part or like sticks to like their little salivary area. It's non-circulative, so it's not actually like infecting the insect itself, but it is sort of hanging out. So they kind of suck it up deeper in here. It's not propagating, but it's like hiding there and waiting until there's another meal. And so that's like semi-persistent. Now you can have it where it starts to circulate, so you see the virus throughout the animal, but it's not actually growing, so it's not infecting the host. I'm sorry, it's not infecting the insect host, but it is sort of like spread throughout the host, which is really interesting. So that's it's circulating, so it's circulative, but it's non-propagative, propagative. Um, and then you've got what would be just normal. Like this is typical for like human viruses too that are vector borne, where it's circulating because it's propagating too. So it travels somewhere in the insect itself and then grows, and that makes a ton of virus, and then the whatever it is, the I don't know, insect can then spit it back out. Okay. So there's lots of examples. There's lots of groups that do research on different aspects, that different creatures, different viruses. Lots of creatures can have different viruses. It just doesn't really matter. So you can see these are three examples. This is actually ignoring this example. Um, but I just thought it was cool because it's got a little bug. It's biting. Anyway, but notice too that its stylus, or stylet, sorry, not stylus, gets all the way down into the phloem and the xylem, and so that can actually transmit through the rest of the plant, which is really, really interesting. This slide, it's a little busy, but the whole point here was just to establish that latency, which is something we've talked about a lot, also exists in plant viruses. And so you can have, over here we have like our plant that is latently infected with some kind of virus, and then how to actually trigger that and get it to transition to active infection is really an interesting acute or active infection. Usually stress is probably going to do that the most, but it's poorly understood. How do you actually get it to transition and get symptoms to show up? Because this can be kind of tricky because you could be dealing with a latent virus and then transmit that plant, and then all of a sudden you have a new infection. And so that's why there are a lot of rules. I just learned a bunch of stuff about the specific quarantine issues and the agricultural check stations that exist in California. So California as an agricultural state is one of the most important places on the planet, let alone for the United States. California as a state makes something crazy. It's like 40% of all the food we eat or something. It's something really high like that. I don't remember exactly, but and so there are really important agricultural check stations. It's kind of like the illegal immigration check stations. It's that same idea. And so like if you're traveling from Nevada into California, those connect, right? They'll have agricultural check stations where it is absolutely within the rights of, this is the video that I was watching, it's within the legal rights of the officers on duty to search your vehicle. They will not call it searching, they will call it looking for plants that maybe they're latently infected or they're transmitting some kind of disease. Because on the books, they decided that is not a Fourth Amendment interruption because they're not looking for illegal things. They're only looking for plant issues or agricultural pests. So which includes more often not viruses so much, but more like insects. because they've decided that it's within it's worth invading your Fourth Amendment rights, which is the right to not have unnecessary search or seizure, um, because the overall damage that these pests can do to our society in general is massive, especially when you have such a strong agricultural state like California, because they have specific examples where it cost the taxpayers like tens of millions of dollars because somebody had an orange in their backseat with something growing on it and they didn't catch it, and it got loose and it wiped out like an entire city's worth of trees or things like that. And so they're like, well, it's worth it. We're not out to find your drugs that you're hiding. We just want to know if there's any oranges or whatever. Anyway, but that's up for debate. But the law is on the side of the officers. Anyway, something interesting to talk about a little bit is the connection between funguses and viruses and plants. So if you've taken a botany course, which I used to teach botany as well, the connection between plants and funguses is very, very strong in a variety of ways. And it turns out that the connection between plants and funguses and viruses is also really interesting. What this diagram is trying to get at is that there are some viruses that can infect funguses and plants, so you get cross-kingdom viral infections. And then, since it can infect both, you can see that in both types of creatures. So you can see it in funguses, you can see it in plants, you can see it in plants that have funguses growing on them or in them, since they have such close-tied connections. So it's super, super interesting stuff. And just to kind of give you a broad exposure to all the things that viruses can do with plants. Now, something we need to briefly talk about is the acquisition of immunity or the presence of immunity. So plants are not helpless. They have a bunch of immune mechanisms. They have innate immunity. So that would be equivalent to our like natural killer cells or macrophages or things like that. They don't have cell-based things. Like they don't have cells like macrophages that will just roam around their phloem and look for viruses or anything. And so they're sort of built into the cells themselves. And so this is a PRR, stands for pattern recognition receptor, which is a consistency that we'll see in human immune systems as well. Basically, they're just looking for foreign DNA or RNA. And if it matches up with something that that it knows is pathogenic. So for instance, single-stranded DNA, for instance, it's not here, like double-stranded RNA, that's really weird, right? You shouldn't typically see that unless you have RNA folding, which, I mean, of course that happens, but like if you see a big old giant thing of double-stranded RNA, that's kind of weird. And so that's probably a pathogen RNA. And so some viruses have double-stranded RNA. And so you have, if you're a plant, you have a pattern recognition receptor that will just bind to double-stranded RNA and initiate one of two immune system possibilities. It's pathogen-triggered immunity and effector. Sorry, I had it pulled up. I keep forgetting ETI. It makes me think of other things, I guess, like ET. I don't know. Effector. So this would be like pathogen-triggered would be like something from the pathogen looks just like that, versus effector would be something that's probably secreted by the pathogen. So like a viral protein itself. So there can be some crossover there, but they sort of mount different immune responses, but there can be some crossover there. It's a whole other world. If you take immunology, you might talk about it, but in this class, we're not going to spend too much time on that. But as far as adaptive immunity, that's a really interesting world. And so a lot like the CRISPR system that you hear a lot about in the news about for doing a gene modifications. So that originated as a bacterial adaptive immune system where it would take viral RNA that the bacteria was able to fight off and take a piece of that viral genome and sort of store it for later. And then when it would encounter the complementary DNA or RNA for that virus, it would attach it into the Cas9 machinery, and then that would sort of act like a little weaponized thing. And so that's it's sort of like RNA interference, which is what plants do. And so they can use pieces of RNA that's designed to attack viral RNA or viral DNA and use that as a weapon. But that depends on the types of viruses that it's dealt with before. And so that's how it becomes adaptive, where it can actually adjust itself. Now, plant immunity can fall into a few different categories. So if you're including these things, these can fall under different categories themselves. And so I just wanted to talk a little bit about that, and then we're pretty much done after that. So you could have dominant or recessive resistance. Okay, so we've got our plants up here, and this is plants over time. So we're sort of following them over time as they get one bite, and then it starts spreading until it's systemic, and then it's just everywhere. Okay, and so the different types of resistance have different time courses, basically. So if you have like a dominant resistance, the level of resistance is on the y-axis, and so the higher it is, the better. And so initially, you can have, like if you have dominant resistance, you have pretty decent resistance early on, so it takes a little bit to actually mount a immune response. But over time, you can actually lose some of that. As far as recessive, there's not really a difference, but when we get into some of our adaptive ones, and so here we've got like RNAi, which that's what I was talking about here, or it uses it as like a weapon. You can see as we start developing, it takes a little time, but the plant starts to figure out, oh, hey, I need to attack the virus using this specific piece of resistant RNA, and then you kind of maintain that, and so you're able to fight it off. Hormone-mediated resistance, that would be like some of the signaling molecules. So you may have heard tell of plants sort of remembering that they're being attacked, or that kind of signaling, or like one plant could be attacked in a colony, and then it secretes uh, like volatile organic compounds that are just basically signaling molecules that tell the other plants, hey, something terrible is happening, I'm getting eaten, or whatever. And so that's kind of what it's talking about here, where you activate these resistances and then it maintains that over time. Yeah, so interesting stuff. The last big topic I just want to talk about, this is a nice review article if you would like to read this. Just talking about all the different things that come together to talk about the push and pull between viruses and plants, and the viral factors and what they actually do versus the resistance genes that also exist. And so just remember that their plants and viruses are constantly co-evolving with each other, right? So a virus can develop a better way to perform translation or to move between cells, and then the plant can develop another way to do that. And so it's just kind of depends on like what's going on and what mutations get engendered. Pretty cool stuff. And the last concept that I want to introduce was just the idea of the technology that goes on. So plants are a great system to use to study things. And so we can study viruses in plants, learn more about them, and then go from there, because it's just easier. It's easier to grow a plant and infect it with a virus than it is even like a mouse, or especially a human, or any other kind of animal. And so we learn a lot about how to actually do applied work in biotechnology using plants. It's a massive field. I knew a couple guys in grad school who were working, because I went to Colorado State, which is a huge ag school, and they were doing the same kind of work that I was doing, only in plants. And so they would study, a variety of things, like how to do PCR on plants because it has different weird things that'll happen, or how to study the genomics and the metagenomics and the quality control for sequencing, and just all this kind of crazy stuff. So biotechnology is a huge, huge deal. There's a ton of money that gets poured into plants and studying plants, and including plant viruses.